So I finished the light dependent, light independent. Let's start from there. I'm going to write down stuff on the board. Can you complete it? Okay, so this is the summary, right? Can you write down the location? And write down the main reaction. What happens in each reaction? Because I need to just go through the light independent again. So you need to remember what we did, right? So this is exactly the same we did in unit 1 and 2. Write down the main inputs, what's going to be the products. The light independent, what is the main input, what's going to be the product. Yeah. So light dependent, the main input is the water molecule. We're going to break it down to produce the energy, right? So I went through this last week. So water is going to split in the presence of sunlight to get the energy to power the independent, right? So energy going to be in the form of ATP and NADPH. But this is the main product. And the oxygen going to be the byproduct. Can you highlight the oxygen atoms? So this is the oxygen. The water molecules oxygen going to be in the oxygen gas. I'm going to ask questions like this. So you have to remember what gives this oxygen, the two oxygen atom, is the water molecules oxygen atom. Right? I'm not going to ask you to rewrite inputs, outputs. We did last week. And what's going to happen in the light independent is the building up of glucose. In the presence of the energy created from the light dependent, right? So we did everything last week. ATP and NADPH coming from the light independent. So you have to know the carbon atom here. Can you highlight the carbon? So carbon dioxide, carbon, one carbon, going to be in the glucose. And carbon dioxide's oxygen going to go into the glucose oxygen. Right? So when it comes to oxygen, it's going to be confusing a little bit because glucose also going to get oxygen, oxygen gas also going to get oxygen. You have to know which one is going to become which. Right, so this is what I did last week. And light independent reaction, also known as Calvin cycle. We just wrote it down like one-way reaction, but it's going to be a cycle, right? So Calvin cycle is something like this, clockwise cycle. So things going to go in and things going to be produced, but it is a cycle, right? So draw a circle. So this is what we're going to learn in detail. So don't look at last all study design questions. You need to know more than that, right? There's lots and lots of concepts introduced. We're going into Calvin cycle in depth, and then C3, C4 chem plants. There's a lot, right? So there's lots and lots of new additions not present in the old one, right? Okay, before I start the Calvin cycle, so Calvin cycle is the same as the light independent, right? It's the same thing. Can you write down, not to confuse with the Krebs cycle? These are the silly mistakes you might do, right? So when I do the cellular respiration, there's another cycle, right? So normally I don't even introduce the Calvin cycle, the name, because you're going to confuse with the Krebs cycle. But now, new study design, we have to introduce the term Calvin cycle for the light independent reaction, but there is a different one in cellular respiration, Krebs cycle. Don't confuse the inputs, outputs. These are two different ones. Okay, before I start the Calvin cycle, I'm going to give questions based on the oxygen atoms and these carbons and things, yeah? Okay, write down the question. So to answer these questions, you have to just look at this slide. Huh? So if I want to track down something, I'm going to label them. So labeling means like something like adding something, like coloring them, so you can track them down. Right? So I can track down different atoms. So sometimes I need to track down what happened to the carbon. Right? So I want to see what happened to the carbon. So I'm going to introduce radioactive compounds. Right? So how we're going to say it is, I'm going to introduce radioactive carbon. So radioactive carbon means it is something like adding a color. So you can track where the carbon going to go. Right? You're tracking down the pathway of the carbon. So if I want to label something, I can use something called radioactive compounds. You can track the entire pathway. Right? It is something like you're coloring the atom, so you can trace the pathway. Right? So it doesn't have to be carbon, you can just introduce radioactive compounds to whatever the atom. Right, so I'm going to just introduce radioactive compounds to different atoms. So these are easy exam questions, huh? So write it down, first one. So I'm going to introduce radioactive glucose. So I'm going to introduce the radioactive glucose. So the radioactivity is going to be on the oxygen atom. Right? So the activity is on the oxygen atom. Right? So I'm introducing glucose, the radioactive glucose with the oxygen atom being labeled. So write down oxygen is the one being labeled. Write down after like 24 hours where you can see the radioactivity. So identify the location or whatever the product that's going to have the radioactivity. Is the question clear? Is my question clear? Right, I'm going to introduce radioactively active compound, radioactive glucose, having the oxygen atom labeled. After 24 hours, you're going to measure the whatever the output, right? So where are you going to see? Oh my God, my question is wrong. Let's rewrite it. I'm going to introduce radioactive labeled carbon dioxide, having the carbon being labeled. After 24 hours, where you're going to see the radioactivity. Sorry, yeah. So let's do the next one. I'm going to introduce radioactive carbon dioxide, the same thing. But now I'm going to label the carbon atom, right? So the same thing, write it down after 24 hours, where you can see what product going to have the radioactivity. So third one, we are introducing radioactive water, having oxygen atom labeled. After 24 hours, where you can see the radioactivity, what is the product? Now it's clear, right? Write down the answers. So only thing is you have to know where this oxygen atom going to be end up in. So carbon dioxide is oxygen going to go into the glucose, right? So that's what I highlighted earlier. So after 24 hours, you're going to see the radioactivity in the glucose, right? Because the carbon dioxide oxygen going to end up in glucose. So same as that, pretty much they're asking the inputs and outputs, huh? So if I label the carbon dioxide's carbon atom, it's going to be still ending up in glucose. So you will see radioactive labeled glucose after 24 hours. But if I label the oxygen atom in water molecule, after 24 hours, you're going to see the radioactive oxygen releasing. Right? I think the confusing one is the oxygen. I can label the carbon dioxide oxygen or water molecules oxygen. Right? So only thing is you have to remember carbon dioxide is the one produce glucose. Water is the one going to break down into oxygen. Right? So otherwise I can ask the other way around. I can say, I have radioactively labeled glucose. That means what type of radioactive compound the plant received. The other way, opposite, right? So to get the radioactively labeled glucose, you need the plants to have radioactive labeled carbon dioxide. Or I can say I can release, the plant is releasing radioactively labeled oxygen. What type of thing the plant received to emit this radioactively labeled oxygen? Right? So if the oxygen being radioactively labeled, The plant needs to receive water, the oxygen atom being labeled. Either way, these are exam questions. Just you have to remember is this one here, right? So water molecules, oxygen will become oxygen gas. Carbon dioxide, oxygen or carbon will produce into glucose. Is this clear? These are multiple choices. Huh? Every year they're going to ask this. Are there any questions? Melissa, you understand? Can't see you, Leonsa, why? Are there any questions? Okay, so shall we start the Calvin cycle? So Calvin cycle is nothing else, it's a light independent reaction. I just wrote the light independent as carbon dioxide building up into glucose, but it is a complex cycle, so we're going to learn the cycle. Can you draw the carbon atoms? I will draw all the carbon atoms, right? So all the circles I'm going to draw during the cycle represent the carbons, right? So carbon dioxide is a 1 carbon compound. It will build up into 6 carbons. So let's draw the Calvin cycle, right? So write down the topic, Calvin cycle / light independent reaction. So if we draw a cycle always going to go clockwise. Don't draw this. But if I draw the arrowhead towards like going into the cycle, they are the inputs. So as an example, if I draw the arrows like this, you're going to follow the arrow head, right? So whatever that's going to go in, going to come out as the products towards the arrowhead. So as an example, if I write ATP, right? ATP is going into the reaction and it's going to be the product, the opposite. Or if in case, if I write the like this. So ADP and PI is going into the reaction and ATP is the product. Right? So you have to know how to read the arrowheads, right? So it's going to go in and coming out as the product, right? So towards the arrowhead, it's always the product. So when you're familiar with this concept, normally they will give either one of these, right? So if I just delete this one, you have to know ATP is the input, so we have to write the output. If I draw like this, you have to know ATP is towards the arrowhead, that means this is the product. So ADP and PI needs to go in. Right, so throughout the Calvin cycle, I'm going to just draw the arrows. If I draw an arrow like this is a representation of a product. Right, so if I draw like this and say glucose, that means glucose is releasing, it's a product. If I draw an arrow just towards the cycle, that means carbon dioxide is going into the Calvin cycle. This is not the Calvin cycle, huh? Yeah, this is not the Calvin cycle. Huh? I haven't included the NADPH. So if it is the NADPH, so if it is going in, you write it like this, and the product's going to be NADP and H plus, right? So, or you can write NADP plus H plus and NADPH, yeah? So, if I write it like this, NADPH is going in, product are NADP plus H plus. If I write it like this, input is the NADP, product is the NADPH. This is not the Calvin cycle. Right, so let's draw the Calvin cycle. So I'm going to draw the Calvin cycle. So the first thing we have to do is the Calvin cycle is going to contain a substrate known as ribulose biphosphate. Right, so I'm going to write it down somewhere. Ribulose biphosphate. You can just write it down RUBP. RUBP is 5-carbon compound. Just remember the carbons. RUBP has 5 carbons. Can you draw 5 circles? So I have RUBP, 5-carbon compound. And this 5-carbon RUBP going to combine with the atmospheric carbon dioxide. Right, so let's draw the arrow towards the cycle. Because carbon dioxide coming into the Calvin cycle. So we're going to say carbon dioxide. Right, so can you represent the carbon using a circle? Right, so the first stage of light independent, the Calvin cycle is the carbon fixation. I'm going to fix the atmospheric carbon into the Calvin cycle. We are introducing an extra carbon from the outside. Right, so each and every circle represents carbons, yeah. Okay, so this is what happened in carbon fixation. We have the RUBP and carbon dioxide. RUBP has 5 carbons. And I'm getting 1 carbon from the outside. So we're going to create something. How many carbons in total we have? 5 plus 1, 6, yeah. So we have 6 carbons. So the product must have 6 carbons. But we're going to split the 6 carbon. So draw how many carbons in one product. It's a simple maths lesson, right? So we are introducing 6 carbons. I took 1 carbon from the outside environment. So we have 6 in total. So the both sides needs to have the same number of carbons, yeah? So the product needs to have 6 carbons, but whatever the 6 carbon product, we're going to split it. So what I'm going to have is a 3-carbon compound, but we're going to create 2 of them. Right, so 3 into 2. Huh? So the name of the product is phosphoglyceric acid, or write it down PGA. I introduce everything in the note. You can read the note, but write down PGA. You can write PGA. Is the carbons clear? But we are not getting only 1 carbon, right? How many carbon dioxides we in the normal photosynthesis reaction? How many carbon dioxide molecules we bring? It's not one, right? How many in the balanced equation? How many carbon dioxides are there? There's going to be 6, right? So can you just times this by 6? So we are using 6 RUBPs and 6 carbon dioxides. Can you work out how many carbons you're bringing into the cycle? It just times 6, right? So RUBP, 30 carbons. And there's 1 carbon in carbon dioxide. And we are bringing 6, so we are introducing 36 carbons. Can you do the math and identify how many PGA molecules you have to produce? So the total carbons we are bringing into the cycle is 36, right? So you have to maintain the 36 carbons in this side. So if one PGA is having 3 carbons, how many total number of PGA molecules we're going to make? I need the carbons, right? I'm not doing a maths lesson otherwise. Just write it down. What? How many? The grade 3 maths. 12, right? 12. Okay, can you write down we are bringing 12 PGA? Okay, I'm going to write it down on the cycle now. We are bringing RUBP 6, and then carbon dioxide 6 of them, and the products we're going to create 12 PGA. Right, so write down somewhere PGA. You're having 12 of them, but each going to have 3 carbons. Right, so PGA is a 3 carbon compound. I don't know what type of questions you're going to get because this is the first time you're doing this Calvin cycle, right? All these carbons and everything, right? I'm going to just do every single possibility, right? So I think it's better to know the carbons, right? This is like easy math. Everyone understand what happened in the carbon fixation? Into the carbon cycle, we are bringing atmospheric carbon, right? Introducing carbon because to build up glucose, we need the carbon. To do that, to capture the carbon dioxide, you need the RUBP. And combining RUBP and carbon dioxide, we are producing a 3 carbon PGA. Is the carbon fixation clear? Right, you do like advanced maths. This is like a simple math, right? So, okay, carbon fixation done. We have altogether 36 carbons, yeah? Okay, let's do the next one. So carbon fixation is the first step of the Calvin cycle. So I have the 3 carbon PGA, but I have 12 molecules, and we're going to break it down, or we're going to just convert it into another molecule. Right, so right on the second stage, after the carbon fixation, we're going to reduce the product reduction. So what happened in the reduction is we are going to create the PGA into something else called G3P. Right, so glyceraldehyde 3-phosphate, if you want to know what it is, it's glyceraldehyde. Phosphate. Right, so it's another 3 carbon compound. So we have just 3 carbon, 12 of them. Can you identify how many G3Ps we're going to make? The balance the carbon, yeah? So we are bringing 36 carbons. You have to have the same number of carbons on the other side. So if the G3P is again 3 carbon compound, so you have to have 12 of them, yeah? So 36 carbons, right? So we are building up, we are creating 12 G3P molecules. Can you write it down underneath? I need the numbers, huh? So we are bringing 12 G3Ps. And from these 12 G3Ps, we are using 2 G3Ps. So if I'm bringing 3 G3Ps, so out of 12, we are using only 2 G3Ps, right? So each molecule is going to have 3 carbons. G3P is a G3 carbon molecule. So you're going to introduce 6 carbons here, right? So we have 30 carbons left because I'm only taking 2 G3Ps. How many carbons in the product? 6 carbons, right? So the 2 G3P out of 12 is going to build up into glucose. Can you see the carbon here? So this is the carbon brought by the G3P. Right, so both sides, the carbon number is the same. Is this clear? Right, okay. So this is what happens in the reduction step. The PGA going to convert into G3P. And out of 12 G3Ps, we are only using 2 to produce glucose. Right, so can you draw the arrow and write down the output? We are building up glucose. Is the reduction step clear? Right, you're going clockwise, huh? So the RUBP and the carbon dioxide, we brought atmospheric carbon into the cycle, and then we converted the PGA, 12 molecules, and PGA converted into G3P, and 2 of the G3P convert into glucose. Is this clear? Okay. So our reduction is done. Let's do the RUBP regeneration. So this is a cycle. We have to keep getting the RUBP again and again and again, because RUBP needs to be an input, right? So only we are taking carbon dioxide from the atmosphere. The cycle needs to produce RUBP within the cycle, right? So whatever the cycle, the leftover G3P needs to go back to the recycling of the RUBP, right? So write down the third step, RUBP regeneration. Right, so the third step of the Calvin cycle is the RUBP regeneration. So the 5-carbon compound always needs to be present within the cycle, right? So the cycle needs to regenerate the RUBP, otherwise you can't do the cycle. Yeah. So what we have is we use 2 of the G3P to build up glucose, but we have 10 left. I don't know how many carbons we have now. So G3P is a 3-carbon compound, but I have 10 left. How many carbons? 30 carbons, yeah. 3 carbon into 10, right? 10 molecules left. So we have the total number of 30 carbons left. Can you build up all the available RUBPs back again? So this is the RUBP regeneration. If the RUBP is 5-carbon molecule, I can make 6 of them. So you're just replacing all the used RUBP back again, right? So because both sides going to have 30 carbon each, right? If we need to continue the Calvin cycle over and over and over, we have to replace the used 6 RUBPs again and again and again. Can you write it down? So in this method, if I use the carbon dioxide, I have enough carbon to replace all the used RUBP, right? So RUBP is the main substrate to capture the carbon dioxide to build up glucose, right, at the end. But you can't keep going on this cycle if the RUBP can't reproduce, right? But if I have 10 G3P left, I have enough carbons to replace all the used RUBP. So we can regenerate RUBP and keep doing the Calvin cycle. Is this clear? Right? Okay, so the reduction step to PGA to G3P, we have to introduce the energy. Can you write down the energy? I'm going to draw the energy, the arrows. Can you write down the correct form? What's going to go in and what's going to be coming out? So the two types of energy, ATP and NADPH, right? So it needs to just go with the arrow. So the reduction step to PGA for the G3P conversion, we have to introduce the energy. ATP going to go in and ADP and PI going to go out, right? Or you can just write down one. Right, NADPH from the light dependent reaction goes in and comes out as NADP plus H plus. Because this is the first time, I'm going to write down everything. No? Is the cycle clear? Okay, I'm going to summarize it. Can you just complete the blanks? Write down the 3 stages of the Calvin cycle. And write down what happens in each. Right, okay. So this is the summary, write it down. right? Can you introduce another ATP here? For the RUBP regeneration, introduce another ATP. We go through the ATP numbers and NADPH later, right? But for this also we are consuming another ATP. RUBP regeneration require energy, yeah. Okay, so the three stages are the carbon fixation, and then reduction, and the last one is the RUBP regeneration. So carbon fixation is, we are taking RUBP, the 5-carbon compound, combined with the carbon dioxide, 1 carbon, and we're going into the PGA, right? So it's a 3-carbon compound. So you have to have 2 of them. So the reduction is PGA, convert into G3P, you can go with the 6s. 12. So 12 PGA, converting into 12 G3P. And out of 12 G3Ps, we are taking 2 of them to produce glucose. Right? RUBP stands for ribulose biphosphate. Just write down RUBP now. Right? It's in the note as well, ribulose biphosphate. Okay, so can you introduce the energy as well? For this reaction, we need to introduce ATP and NADPH. And then comes the leftover 10 G3Ps. Going back to recycling the RUBP. Yeah, you can balance the carbon, right? So 3 carbons, 30 carbons, and we have 5-carbon compound, 30 at the end. Right, so this is the most important thing. We have to replace the RUBP back again. Otherwise, you can't keep doing the same cycle again and again and again, right? The Calvin cycle will stop if you can't regenerate RUBP. So make sure you balance in the carbons. Right, for this one also you need ATP, right? So Calvin cycle needs lots of energy, huh? It's energy consuming. Is this clear? Okay, right, so Calvin cycle is this. This is what you have to know for VCE, right? But for the carbon fixation, to bind the RUBP and the carbon dioxide, there's a special enzyme involved, right? So this is the most abundant enzyme in the whole world, which is known as Rubisco. So can you write down the enzyme on the top of the arrow? Just write it down, Rubisco, right? So Rubisco is the enzyme that's going to catalyze the carbon fixation. So you're learning enzymes under this photosynthesis. This is where the enzyme gonna come, right? There's only one enzyme we're going to teach you, the Rubisco only. But if this is the Rubisco, so it's just going to just go with all the factors we learn, right? So Rubisco is going to be affected by the temperature, the pH, the substrate concentration, everything, right? Just write it down like this. U simple, IS simple, Rubisco. Right, rubulose 1,5 biphosphate carboxylase oxygenase. No need, just write down Rubisco. Right, so this is what the Rubisco stands for. But Rubisco is the enzyme involved in this process, right? So can you draw the enzyme and the substrate diagram of binding and forming the product? The same exact thing I did with the enzyme. You have to identify the substrate, substrates, and the enzyme, and then draw the product, active sites and everything. Huh? So I'm going to write down only the carbon fixation, right? So this is what the reaction is, right? So you can see the substrate, you can see the enzymes. Can you draw the product formation? Exactly the same thing we drew under enzymes. Draw the enzyme. Rubisco is the enzyme. Right, so this is the enzyme with the active sites. And the two substrates are RUBP and carbon dioxide. Right, so the two substrate bound to the Rubisco's active site, creating the product. Draw the product formation. Think about now the Rubisco going to be used up or not, and draw the product. So Rubisco is the enzyme not going to be used up in the reaction. So whatever that's going to go in needs to come out as it is. Right, and then draw something for the PTA. Right, you can just draw the like this or whatever the shape, yeah. Can you draw the carbons underneath? I'm going to do something else without the carbon dioxide. So you have to understand this carbon thing, right? So if you understand this, can you draw the carbons underneath so we can balance it? RUBP, remember, as a 5-carbon compound. There's 5 carbons to RUBP. Carbon dioxide bringing additional extra carbon to the cycle. And we're going to just combine these two, introduce it into the cycle. Right, so this is what happens in the Calvin cycle in the presence of Rubisco enzyme. So if there is a plant, the green plant, they are undergoing the Calvin cycle. So at the end, they can produce glucose. Right? So glucose is the end product of the photosynthesis. So the plants can use it for the cellular respiration. Right? So if the plants doing the photosynthesis, only they are doing it to produce the glucose, to break down to produce their ATP. Right? So Calvin cycle, the last final, the most important product needs to be glucose, nothing else. But there is a problem. The Rubisco will bind with atmospheric carbon dioxide only if the carbon dioxide concentration is really high. Right, so Rubisco is an enzyme. It can bind with the carbon dioxide if there's lots of carbon dioxide available. Right, so if there's no carbon dioxide, enough carbon dioxide, Rubisco will bind with the oxygen. Right, so instead of carbon dioxide, let's see what will happen if the Rubisco binds with the oxygen. So write it down. I'm going to do the same thing, but without carbon dioxide, it's going to replace with oxygen now, right? So if there's a lot of oxygen available outside in the environment, the Rubisco will tend to bind with the oxygen, not the carbon dioxide. Right, so the same substrate RUBP, but instead of carbon dioxide, we have only oxygen left. Can you see this is not introducing that extra carbon into the cycle? Right, there's no extra carbon here, right? So that means if you think about the entire cycle, they're not going to have enough carbon at the end to regenerate the RUBP. Right, because oxygen is not bringing a carbon to the cycle. If the oxygen is not bringing the carbon to the cycle, they can't continue the Calvin cycle. So without continuing Calvin cycle, there's no glucose production. Right, so can you draw the same thing? RUBP binding to the oxygen with the Rubisco enzyme. Do exactly the same thing, but you're not introducing carbon dioxide, now instead oxygen. Right, so this is the Rubisco enzyme. RUBP is exactly the same thing. But we don't have carbon dioxide, instead we have is oxygen. Right, so oxygen doesn't have that extra carbon that the carbon dioxide will have. So without that extra carbon, we can't continue the Calvin cycle. So no Calvin cycle, there's no glucose production. There's no glucose production means the plant can't produce energy. Right, so the previous one is the Calvin cycle, but this is something else. If the Rubisco binds with this oxygen instead, it's known as the photorespiration. Photorespiration is bad because it's going to stop the Calvin cycle and it's going to stop the glucose production. I will send you a document so you can compare the Calvin cycle versus photorespiration, right? So you know the entire cycle of the Calvin cycle, and I have a document which is going to show the photorespiration, right? You can count the carbon and try to understand it, but all I want to say for the photorespiration is not bringing this carbon dioxide, the carbon from the carbon dioxide to the cycle. So it won't have enough carbon to recycle the RUBP. It can't keep doing the Calvin cycle, right? So if you have like the chemistry knowledge and anything, you can balance the carbon and then understand the photorespiration, right? But for VCE, I don't need it, but I'm going to give the document so you can have a look. Is that clear? But the only thing you have to remember is photorespiration is bad because that's not bringing the enough carbon to maintain the carbon Calvin cycle, right? There's no glucose production, so plants don't need the photorespiration. It's a bad process, right? I'm going to send that document. You can have a look after the class. I'm going to share the document. I printed out the notes before I add this, right? So it's not going to be in the note. Can you take a screenshot? I have printed out the note, so you don't have it in the note. Huh? And then I realized you need to know this. So I just added it in. But take a screenshot. This is it. Can you see the comparison between the Calvin cycle and the photorespiration? Photorespiration is bad because it's not having enough carbon to recycle the RUBP. Definitely it lacks that 6 carbon, right? Carbon dioxide, 6 molecules not coming. So you can't regenerate the RUBP and there's no glucose production. Is this clear? So there has to be 10 T3Ps to recycle, right? If there's no enough carbon, you can't regenerate the RUBP, right? Without RUBP, there's no the Calvin cycle or there's no glucose production. Right? Everyone took the screenshot? Okay, so this is a huge problem for the normal plants, right? So if the plants like live in normal condition, this is something that can take place. So let's do the stomata. Can you remember the stomata? So if this is the cross section of a leaf, right? So we're taking if this is the leaf, I'm going to take a cross section. And this is the little hole or the stoma that's going to just help for the gaseous exchange, right? So carbon dioxide going to go in, oxygen will go out through the stomata. Right, many stomata, one is stoma. So let's say when the carbon dioxide, during the daytime when the stomata is open, carbon dioxide going to go in, right? So there's inside the leaf, there's lots and lots of carbon dioxide. But at the same time, there's going to be oxygen produced as well. So oxygen needs to leave through the stomata. But what happens is, if the outside is really, really hot, if there's lots and lots of like high temperature, if the temperature is increasing, what else going to go through the stomata is the water, right? So you learned this water evaporation last year, right? So the transpiration and pull and everything. So the stomata is not for the water evaporation, but if you have like an open hole, the water can evaporate through the stomata at the same time. But if there's like a big temperature difference, there's lots and lots of water removing from the plants. The plants has to shut the stomatas to stop the water evaporation, otherwise they can't survive, right? So at one point, if the outside temperature is very, very dry and hot, the plant needs to close the stomatas, right? Because they need to save the water. But what happens if the stomata is closed? If the photosynthesis is keep happening, they will use up all the available carbon dioxide. Right? In the Calvin cycle, the plant will use all the available carbon dioxide. But at one point, they will use up most of the carbon dioxide, and the Rubisco won't have anything else. They will have high concentration of oxygen left, right? So in this case, the plants will switch from Calvin cycle to do the photorespiration. Is this clear? Right, so this happens in like normal plants. So if you take like the plants, like the tropical plants, this is a huge problem for the normal plants, because if the outside temperature fluctuates, they will shut their stomatas, and if they use up all the available most of the carbon dioxide, the plants will switch from Calvin cycle to do the photorespiration instead, right? So photorespiration is bad. It doesn't provide any glucose. It just consumes ATP. Right, so write down what the normal plants are. So I use the term normal plants. Normal plants means if you think about like a normal plant around like Melbourne. So what the normal plant's going to do is, during the Calvin cycle, the first stable compound they are going to produce is the PGA. Can you go back to the cycle and highlight PGA? So this is the Calvin cycle. This is the start of the Calvin cycle, the carbon fixation. So during the carbon fixation, we're going to use the carbon dioxide, RUBP, and create the first stable compound. Can you highlight it? So PGA is the first stable compound in this normal plant, right? So the first stable compound they're going to create during the Calvin cycle is a 3-carbon compound. So we're going to call these normal plants as C3 plants. Is this clear? Right, so don't use the term normal plants, they are C3 plants, right? So C3 plants are nothing else. During the Calvin cycle, they're going to produce the PGA, a 3-carbon compound, as the first stable product. Right, so these plants, the C3 plants in the normal environmental conditions. But if the temperature fluctuates and if the temperature becomes very hot and dry, they have to experience the photorespiration, because high and dry temperatures, high temperature, dry environment going to just increase the transpirational pull or water evaporation. So to stop the water evaporation, they have to close their stomatas, right? So write down normal plants are C3 plants. So the C3 plants will experience the photorespiration during a hot and dry day. To stop the water evaporation, they will close the stomatas, and the photorespiration will use up most of the available carbon dioxide, leaving on the carbon oxygen left. And the oxygen will bind with the Rubisco enzyme and undergo photorespiration instead of the Calvin cycle. Photorespiration is not producing any glucose, but just using the ATP or the energy. So the plants needs to do something to avoid the photorespiration. But normal plants not adapted to stop the photorespiration because they're not experiencing this every day, right? That's why I said normal temperature plants, right? So if there's like a sudden temperature hike, they can't do anything. But if in case plants are living in like totally in like the very hot environments, like the dry desert environments, they have adaptations to stop the photorespiration. But C3 plants not going to have any adaptation because they live in normal environments, right? They're not experiencing the photorespiration every day. But if a plant is permanently living in a hot and dry environment, like the desert plants, they must have an adaptation. Is this clear? Right, so the C3 plants will, not every day, but they can experience the photorespiration because they don't have any adaptations to stop the photorespiration. But if you consider the plants that permanently living in hot and dry environment, they must have adaptations to avoid photorespiration, otherwise they can't survive. Right, so the normal plants, I said C3 plants. So C3 means the first stable compound, PGA, is a 3-carbon compound. Right, so is the C3 clear? So shall we draw the plant leaf of C3 plants? And then I'm going to compare it with the dry environmental plants. Right, so the plants permanently living in dry environments, hot environments. Right, the cactus, something like that. So the normal plants are C3. I will introduce the dry plants as well. It's not C3, it's C4. Shall we draw the cross section of the plant leaf of the C3 plants? So C3 means the normal plants, right? So write down if you want, I will give you the name of the plants. I keep saying normal plants, C3 plants. Everything in the note. Now you have to read the note. Potato. Sugarcane. Potato is a C3 plant, normal plant. Oats, sugar beet, barley. Sugarcane is a dry, hot environment one. C3 is potato as an example. Right, so C3 doesn't have any adaptation. They have to experience photorespiration. They don't have any adaptation. So I'm going to draw the C3 plant, the leaf structure. And then I'm going to use these terms next week. So you have to know the parts of the leaf. I'm going to change the colors, use different colors, and then draw this. Huh? So if this is the leaf, we're taking a cross section, right? But these ones are C3 plants. So the first stable product created during the Calvin cycle is the 3-carbon PGA, right? So that's why they are C3 plants. So if you consider a plant, the top layer is always a shiny layer. What is the name? cuticle. Right, so plants needs to have a cuticle, but needs to be something made out of lipids. Right, so the top layer of the plant is the cuticle, but this cuticle is made out of a lipid known as the waxes. So we call it as the waxy cuticle. Waxes going to make the leaf, the top part, shiny. The normal wax, right? So normal wax is a lipid, but you can have like a layer making the cuticle. So the presence of the waxy cuticle is going to reflect all the additional extra sunlight, as well as it's going to just stop the water evaporation. So waxy waxes means something water insoluble, right? So waxes are lipids. Lipids don't going to let the water evaporate. And just underneath the waxy cuticle, you're going to see something called epidermis. So draw like compact, like a brick layer, one after the other. This needs to be tightly packed to stop the water evaporation again. Right? Plant is doing everything to save water. Right? So this is a tightly packed brick-like cell, which is known as the epidermis. So why they've been tightly packed is again to prevent the water loss. Right, so write it down. Epidermis is also going to prevent the water loss. So these two cell layers going to be present at the bottom of the cell as well. So can you draw a thin cuticle and an epidermis again at the lower surface of the leaf? So you can make the lower one like less thick, the cuticle, the upper one needs to be thick, right? So because the upper surface is the one that reflects the sunlight or exposed to the heat. So the upper surface needs to have a thick cuticle than the lower one. Can you label the epidermis? There's one at the top and there's going to be one at the bottom. So the top one is the upper epidermis. The bottom one is the lower epidermis. Right? If you want, you can say upper waxy cuticle, the lower waxy cuticle, right? But still, none of these cell layers contain any chloroplast, right? So the topic is the photosynthesis. None of the cells contain chloroplasts. So the next layer I'm going to draw is something like this shape, elongated cells, use the correct shape. Huh? So they are longer than the brick layer, right? So longer than the epidermis. But they need to be tightly packed like this. So can you arrange the cells one after the other? So this cell is known as the mesophyll cells. And they're going to have chloroplast. This is very important. Huh? You have to know this to understand the next week's lesson. You have to know where the chloroplasts are, right? We're doing the C3 plants. C3 plants going to have the chloroplast inside the mesophyll cells. So dot, like all the cells with the dots, dots represent the chloroplast. Yeah. So we have the cuticles and we have the epidermis, and then we have like a tightly packed columnar shape, like columns, tightly packed mesophyll cell layer, and just beneath this layer of mesophills, you have another loosely packed mesophyll layer. Can you draw the second layer, mesophyll layer? But there's going to be lots and lots of space in between the second layer. Right, so both mesophills will contain chloroplast. Nikita, clear? So you can't just name the both layers as mesophyll. So the first layer with like the elongated cells known as the palisade mesophyll. And the second one with big air pockets known as the spongy mesophyll. But whatever the mesophyll, mesophills will contain chloroplast in C3 plants. Is this clear? Okay, so C3 plants will contain mesophills in the palisade and the spongy mesophyll layers, right? Can you write down the difference between the palisade and the spongy mesophyll? What you can see? So the palisade cells tightly packed, right? But the spongy mesophills Can someone tell me what is this space here? Intercellular or intracellular space? I'm highlighting here. Which one is it? Inter or intra? Send me the answer or tell me the answer. So that's what you have right? There's lots and lots of spaces in between the spongy, but not in the palisade, right? But you have to use the correct term. What is this space? Intercellular or intracellular space? What is it? Remember we did inter-intra in homeostasis? Tell me the answer.
Miss, is it intercellular?
It is intercellular space. In between the cells, always inter, right? Not intra. So the spongy mesophyll is different to the palisade because there's lots and lots of intercellular spaces. These are the keywords you have to use. Huh? So large intercellular spaces present in the spongy mesophyll, absent in the palisade. And you can see the compacted cells in palisade, but loosely arranged with lots and lots of intercellular spaces with the spongy mesophyll layers. Okay, we forgot the stoma. Can you just delete one of the epidermis and the cuticle? And we have to introduce the stoma, the stomata. Right, there has to be a gap between the epidermis and the cuticle. So this is the space controlled by two cells. So this is the space that's going to let the gases to exchange. At the same time, the water going to operate. Singular is stoma, plural is stomata. Otherwise, just write down stomata. So stomata can be opened and closed by the action of these two guard cells. Can you write down the name of the cell? Guard cells. Right, so the guard cell is going to open and close the stomata. But why do you think the spongy mesophyll going to have lots of air pockets? Why the spongy mesophyll going to contain lots of large intercellular spaces? Right, so this is the intercellular space. Whatever the gases that are going to come in can be stored in these intercellular pockets. Is that clear? So the stoma is the one that's going to let the carbon dioxide in, right? So once the carbon dioxide comes in, there has to be a space to store the carbon dioxide. Or once the oxygen produced, there has to be a place to keep the oxygen. Right, so there has to be pockets to keep the oxygen and carbon dioxide. Right, so the C3 plants will have the spongy mesophills with a lot of intercellular spaces, right? Intercellular spaces are the air pockets. After the class, draw this leaf structure. I'm going to do the C4 as well. Huh? Okay, so the last one is, remember the structures that transport water and food in plants we did last year? xylem and phloem, right? So we have to introduce the xylem and phloem. I don't know whether how much you remember from last year. I told you to do 1 and 2 properly. Vascular plants, vascular bundles. Remember we did something called vascular bundle? So vascular bundle is made out of 2 things, the xylem and phloem. Xylem is to transport water. Tell me what the phloem function is without using the term food. What is the specific substance that transported by the phloem? If you're not sure, you can write food, yeah? But it is not food, there has to be something. It's not even glucose. It's sucrose. If you don't remember, don't write sucrose, just say food, that's enough, right? We're not doing 1 now, unit 1. But vascular bundles made out of the xylem and the phloem. And it needs to be surrounded by a bundle sheath cell. I'm going to introduce the bundle sheath cells now. Can you highlight the bundle sheath cells? This is very important for this chapter. So I'm going to draw a vascular bundle like a circle. And inside the circle, you have the xylem and the phloem. Right, so this is what we draw as the vascular bundle last year, but vascular bundle needs to be surrounded by bundle sheath cells. Can you draw lots of bundle sheath cells like this? So they look like the bricks, one after the other, compacted bundle sheath cells. Can you label them? This is the bundle sheath cells. Go and redraw the same thing in the inside the leaf. For 3, 4, you don't need xylem phloem, you need the bundle sheath for this one, right? Is this clear? Netpi? Momal? Gopika? Can you draw the exact same thing here? Can you put the vascular bundle somewhere here, the circle? Arrange everything. I need the bundle sheath, I need the xylem, I need the phloem. right? So this is it. This is the cross section. Okay, if you understand this, write down this question. Okay, so in C3 plants, the photosynthesis will take place in whatever the cells. Write down the answer. Finish. So you can say mesophyll or you can say palisade and spongy mesophyll, right? Everyone understand the C3 plants? So I said C3 plants are the normal plants, right? In normal environmental condition. They don't have adaptations to stop the photorespiration because if there's a sudden temperature hike, they can't do anything, right? But there is a special adaptation in the plants that living in permanently in hot and dry environments. So they will have the normal Calvin cycle, but during the Calvin cycle, the first stable product they're going to produce is not the C3PGA. The first stable compound is a carbon 4 compound. Right? So the plants adapted to live in dry and hot environments known as C4 plants. I will introduce the whatever the compound next week, oxalic acid. It's a carbon 4 compound. Right? So there's two types of plants, the C3 versus C4 plants. C3 produce the PGA as the first stable product, but C4 adapted to hot and dry environments produce something containing carbon, 4 carbon compound. Right, so because I just drew the C3 plant, the plant leaf cross section, I'm going to draw now the C4 plant, the cross section. But remember, these are the special adapted plants just to live in hot environment. So they are doing the best to minimize the photorespiration. Right? So these plant leaves are adapted to minimize the oxygen intake or to minimize the oxygen binding to the rubisco enzyme. So I'm going to introduce the things only. So the only difference between the C3 and C4 is they're going to have a bigger bundle sheath and a vascular bundle. And then around the bundle, the vascular bundle, they're going to have lots and lots of mesophyll cells arranged, right? So let's draw the C4 plant. So there's no difference. The cuticle, upper epidermis, the lower epidermis, the same. So can you draw the C4 plant still again? So the normal cuticle, right? You can leave the gap for the stomata. and draw the epidermis upper and lower quickly. Right, so these are not the normal plants, they are specially adapted to the hot and dry environment because they are permanently living in the environment. So if the photorespiration happens, it will happen always, then they can't survive, right? They must produce glucose. So can you draw a vascular bundle? A big prominent vascular bundle. Can you highlight the bundle sheet cells? And chloroplast going to be present in the bundle sheet cells. Can you put the green dots inside? And then I'm going to draw the mesophyll cells. There's no two types of mesophyll cells, you have only mesophyll cells. But it's just going to arrange towards the bundle sheet cells. So I'm going to draw the mesophyll cells clustered around the bundle sheet cells. Right, you're packing all the mesophyll cells around the bundle sheet cells. Is this clear? The two most important cells for the C4 are the bundle sheet and the mesophyll, but the bundle sheet are the most important. Right, so mesophyll will have chloroplast as well, plus in the bundle sheet. Right, so the rest has to be mesophyll. Huh? Can you write down this statement? Mesophyll cells clustered around the bundle sheet cells. So you did not see this in C3 plants, right? So these are specially adapted to avoid photorespiration. So they have the bundle sheath cells containing chlorophyll, chloroplast, plus the mesophyll cells, but the mesophills clustered around the bundle sheath cells. I'm going to send you the leaf document. If your diagrams are not good, you can keep my mark. I'm going to send the document. It has the C3 cross section and the C4 plant cross section. Everyone got it? So you can see the beautiful one. Everyone got this document? So you can see the C3 to C4 comparison, right? So C4 is special. You will have bundle sheath cells, but the mesophyll will cluster around the bundle sheath cell, right? So you can't see a similar one in C3 plants because C4 is specially adapted to avoid the photorespiration. Right? There's no bigger gaps, right? The intercellular spaces, there's nothing. But next week, if I use the term bundle sheath, can you add bundle sheath the chloroplast? It doesn't have in this one. You have to have the green dots in the bundle sheath, yeah? But absent in the C3 plants. But the bundle sheath cells will contain chloroplast in the C4 plants. Plus, it's going to be present in the mesophyll cells. So if I use these terms, you have to be familiar with the terms, right? Bundle sheath, the mesophyll, the palisade, the spongy mesophyll. Is that clear? So C3 plants, the entire photosynthesis will take place in the mesophills. But C4 plants, they're going to swap between the mesophills and the bundle sheath cells, right? To avoid the photorespiration. So I'm going to give a name for this structure. So you wrote down this, right? So the mesophyll cells clustered around the bundle sheath cells. That means this section here. So this section here is known as something called, write it down, Kranz anatomy. Right, so Kranz anatomy is nothing else. You have lots of mesophyll layers clustered around the bundle sheath cell. You can't see a similar one in C3. This is an adaptation to avoid photorespiration. Right, so this structure is the Kranz anatomy. Kranz anatomy is absent in C3, only in C4. Hurry up, I have questions as well. Can you compare the C3 and C4 plant leaves? Do this comparison quickly. Identify the cell types that are going to have the chloroplasts. Write down whether the intercellular space is present or not. Present or absent, you can put the ticks, yeah? And then which one going to have the Kranz anatomy and which one doesn't have the Kranz anatomy. Second and the third, put the ticks. First one, write down the location. Right, this is only thing you have to know for next week, C3 versus C4 plant structures. Okay, I'm giving homework. I'll give you 2 pages to do, right? The questions for photosynthesis, light dependent, light independent, C3, the plant leaves. Yeah, I haven't done the photosynthesis different types, right? So what you're going to learn is the C4 photosynthesis and something called CAM photosynthesis, right? So what we have done at the moment is the Calvin cycle for the C3 plants, right? So this is the normal C3 Calvin cycle. But I will do the C4 and CAM photosynthesis next week, but you have to know whatever we learned today, right? So the structures and everything, I can't go through them again. So if I use the term bundle sheet, you have to know where they are. If I say mesophyll, you have to know where they are, right? These are very, very important. No? And then Cran's anatomy, and then all the adaptations and stuff, right? So I'm going to send 2 documents. You can do them. So if you do it, ask for the question answers, I will give the answers. But you have to do them first. I have one. So first one I sent, light dependent, easy questions. Second one, C3 plant structure questions. Everyone got both documents? Right? Yeah, Akshara and I'll email. Huh? If you want me to email, let me know. Akshara and I need your email. Send me the emails. Jinuka, you got it? Homework, huh? Can you do them all? Do some work. There's a lot of content, right? If you haven't done 1 and 2, there's a lot of things to learn. Do them. This is a big chapter. It replaces the cell signaling from last year, right? So a lot of content. Everyone got it? Then you can leave if you don't have anything. Bye.
Thank you. Bye.